Hi, everyone. We're just going to give folks a few seconds to get logged in, and then we'll get started. All right, I think we're starting to level off here. So why don't we go ahead and get started? Before we begin, let me just note that this session is being recorded and the video will be on our website after today. So just to be aware that it's being recorded. My name is Liz Langdon-Gray, and I'm the Executive Director of the Harvard Data Science Initiative. It's my privilege to welcome all of you to our final industry seminar of the academic year. We are delighted to see so many of you here today. I think it's fitting that our final speaker, Arwa Dilaimi-Wendler, is from one of our closest corporate friends, Bayer. Bayer is a corporate member of the HDSI and a major supporter of our recently launched project on trust in science. We've enjoyed working with our colleagues at Bayer to explore fundamental issues at the nexus of data science and society. But today we have the opportunity to hear about how Bayer thinks about innovation in data science in support of their core activities in R&D. And we are delighted that Arwa will be sharing her experience with us. Arwa is a data scientist and project lead at Bayer Crop Science R&D, focusing on disease control. Arwa holds a PhD in bioinformatics in industrial biotech, an MSc in systems biology, and a B in genome research. In 2019, she was recognized by Bayer with the Distinguished Women in Data Science Award, and since 2020, she has co-led the Bayer Europe Network for Women in Data Science. We're so pleased that she's here with us today. She's going to talk about her experience at Bayer, give you some insights into innovation in data science at the company. And we're going to have her share her screen, give her talk. Please feel free to submit questions using the Q&A feature as she speaks, but we're going to save answering those questions until the end of the presentation. Arwa, thank you.
Thank you for inviting me to let me share my screen first. So, thank you and good morning or good evening to everyone joining in today. Thanks also from my side to you for your interest and the invitation by the Harvard Data Science Initiative organizers and Jonathan Geibel from the Bayer Life Hub in Boston for inviting me. I am going to give you today some insights into my work as bioinformatician and innovation project lead in data science at Bayer Crop Science. But before I go into the details, I would like to give you a brief introduction into my personal, academic, and professional background, even though Liz already gave some insights. So starting with my personal background, I get often the question, where are you come from? Mostly I get confused and say from planet Earth. But actually I traveled a lot during my childhood with my parents and lived in different countries. I was born in Jordan, lived in, have the Iraqi and the German citizenship in the meantime. lived in Algeria and before moving to Germany, finishing school and going to university, where up to my PhD, I was focusing my research mainly on bioinformatics, genome and systems biology around epidemiologic and virulence characterization of human pathogens like ulcerans, diphtheria, and cholera. During my PhD, I changed use case-wise the focus towards bacterial industrial production of various compounds from glutamate to vitamin Bs. Interesting, it wasn't such a big shift as you may imagine, since the organisms leading to these illnesses and those beneficial ones, for example, for vitamin production, are very closely related. From my professional career-wise, I started in the academic field as coordinator for all tools and platforms at the German Network for Bioinformatics Infrastructure that provides chem and bioinformatics resources, databases, and compute infrastructure as service, but also training to the entire national German academic network. As often at academia, you end up with doing different jobs at the same time. Here I was a bioinformatician in addition in different projects, from academic to industrial to even governmental organizations, like the German Army forces. It was the time there, it was really an eye-opener to see the variety of use cases where you can apply bioinformatics and systems biology. In 2017, I accepted a position at Bayer in the small molecules R&D organization, where I was first in the team responsible for overseeing the data science technology landscape that we have within R&D and identify the potential gaps and fill them with internal and external innovation. Later, I moved to focus back on bioinformatics, specifically around pest control, and recently joined the disease control team. Before I go into my activities in detail with bioinformatics, I would like to give you a brief introduction into the focus areas of Bayer and where my work as bioinformatician plays a role here. Bayer has, as you may know, 3 main operational focuses: consumer health, pharmaceuticals, and crop science. Consumer health focuses on providing diverse, very well-known over-the-counter, so prescription-free products for everyday health, like Bepanthen or aspirin. Pharmaceuticals is focusing on prescription-based medicine, like for cardiology, oncology, gynecology, and so on. But they are focusing also on newer topics like cell and gene therapy, or collaborations with external companies like CureVac for corona vaccination production. At Crop Science, where I'm working, our mission is quite bold. Our mission is to shape the future of agriculture. I have to say, working now since 4 years at Bayer, I am realizing where we are all innovating and shaping the future of agriculture. And I will showcase you several examples in this presentation. Why does the world need more innovation in agriculture? By 2050, the demand for more food, feed, and biofuel will increase by 50%. The world population will rise to 10 billion human beings, and they need all to be fed. And 70% more meat will be needed in developing nations. While we have on the one side increasing demand, we have on the other side around 17% harvest loss by 2050. Further, we have 12 million hectares of agricultural land we are going to lose annually, and the arable land per person will reduce also by 20%, mainly due to climate change. That's why we need innovation that meets the needs, the needed increase of produce while protecting our natural resources, and we need to produce more with less and less with the time. How does crop science want to achieve this goal? So our mission, our vision is to develop tailored solutions for our customers that brings the right combination of for each customer of innovative seeds and breeded traits, crop protection during the entire season, and digital solutions for forecasting and monitoring during a season. And especially before even a seed is planted. Each season, farmers make hundreds of decisions that impact their bottom line, their yield, and their entire existence. That's why farmers need to use all the information, data, tools, and technologies available to make the best decisions in every step they make. Please don't underestimate the digital maturity of modern farmers today. They are highly digitally fluent and very well knowledgeable and connected. Every step they make on their field is tracked and translated into data that is processed through a combination of different factors, like using sensors to detect and manage water and even nitrogen level. And they require, of course, a partner that streamline their data and unify uniting these data with machine sensors and even satellite data to generate harvest maps, as you can see here on the right side. And predict, depending on the upcoming weather, in everything in one centralized hub. So it makes it user-friendly for the farmer. This is done by our climate cooperation colleagues mainly with field view as solution. I hope you can see here this upper one. Yeah, banner. Okay. Yes. So how does it look like at the end? So one of the best examples of a result of how these advancements are made an impact for the farmers and our customers, of course, we see today with corn. Today, we see a huge increase of efficiency of producing actually the same yield of corn, but with a fraction of the size of farmland that was needed in 1940. This is an example of how we can produce more with less input, in sum for the farmer, less space, less work, and less investment. As mentioned, within crop science, there are three further operational layers or pillars: crop protection, seed traits, and digital farming. And in my position, I am in R&D. I am data scientist in disease control within crop protection. What are our goals in crop protection? We are discovering and developing products that protects the plant at each level of their development, from the seed up to the healthy plant. For seeds, for example, we are developing different coating solutions that protects the seeds from pests and nematodes during the transport to the farmer, the storage, but also in the soil when they are planted. Herbicides for weed control, that especially weeds take place and nutrition away from the actual crops, which is of high relevance, especially due to the high speed of weed growth compared to the crop, the actual crop of interest. Without an effective weed management, especially against resistant weeds, the farmer might end up with basically burning down the entire field and losing the entire season yield to avoid the risk that such weed resistance remains on the field during the next season or move even to neighboring fields. Similar situation also for insect control and disease control. And that's why the farmers need to understand very well the history of their field and the actual development in real time to overcome such issues as early as possible and to make the best decisions. Okay, So what is the process to identify new products, new candidates for agrochemical crop protection? Let me sum it up. It's for sure not that easy as you might think. First of all, we need to screen every year hundreds of thousands of new compounds for their activity against our target organisms, but that needs to be safe for the crops, of course. Further, so of course, as you can imagine, also during the last years, there are not so many compounds that not have been tested yet. So it's crucial to design from the scratch entirely completely new chemical compounds that can be tested. So we need to design new compounds through virtual screening and modeling of desired properties using machine learning and deep learning. And here we need to join forces. And here we join also forces with colleagues not only at crop science, but also with pharmaceuticals and consumer health to optimize jointly the models we have at hand to reach better accuracy for the prediction of our respective required properties. As you can see here, the investment is also very high, and we are investing of around 300 million of euros for one compound that reaches the market. And it takes up to 14 years that a candidate end up on the market. So it's a huge investment, money and time-wise, and resources, people. So it's very crucial that we start at the beginning with good compounds that have higher probability to achieve the next and over next steps. So besides activity in the target organisms and safety to the crops, we need also to identify if the compound is safe for the humans and beneficials. This is a main cornerstone of our activities, aside of activity, of course, in the target organisms. Also, we need to find out, is it safe for the environment? So we are testing how deep does the compound penetrate the soil, and if it's degraded, how its products are behaving in the soil and also in the groundwater. Does it reach the groundwater? Such things we have to analyze and understand the threshold. Yes. So within the R&D pipeline, we are generating also a huge amount of diverse data types from molecular level in genetics and chemical experiments up to satellite imagery worldwide. All these data require a combination of advanced technologies for generation, adaptive IT infrastructure for processing, on-edge data analytics, and integration of data from various sources and technologies for use for different use cases, which ranges from target discovery, so finding new molecular mechanisms that are active in the target organisms, but not active in the off-target organisms. These understanding the resistance development, also up to tailored solutions for our customers, product recommendation and precision planting, but also output-based pricing, so that the farmer doesn't pay more than we can deliver to him at the end with his yield. So at each step, so this is basically the start, and here's where it ends up. We are applying predictive models to predict the outcome of the next step, and even this step over next step. And we are applying also data science to learn from past experiences and from the next experiences to optimize the portfolio of the projects and the actives we have in the earlier research. However, since you might be more familiar with the pharmaceutical field than the crop science field, I would like to make here in the following a comparison between pharmaceuticals and crop science, and to showcase you what are the differences and what are the overlaps. So the main goals of both organizations and both activities is to find new candidates that have innovative biological mode of action. Especially finding new mode of action is even harder than finding new candidate, because that happens once, maybe every 20 years. So if you find a new mode of action, let us know. So at Pharmaceuticals, as said, they have the same goal, but they are focusing on human. Everything they are doing is human-centric. Here you need to find if the compound is active and bioavailable in the human organ where it should be active. And of course, it doesn't have to be toxic against the human. They need to find out how the synthesis and the upscaling works for production, and to test all these in clinical studies. Where in human, there is a lot of information also available externally in academia. At crop protection, we need to generate a lot of our own data from the scratch. And this is quite complicated, especially because we are not focusing on one target organisms. We have so the compounds and candidates we are identifying, they need to be, of course, active against specific organisms like insects, diseases, and weeds. And this is very generic terms. So there are hundreds of species which lie behind these terms: insects, diseases, and weeds. But they have also to be not non-active in off-target organisms, like, of course, human. It has to be environmentally safe. And with environmentally safe, I mean we are to the environment, so the soil, the water, and so on, but also to organisms living in this environment. So we are testing the activity from the waterlands up to birds and animals, how active, if we have activity there. And of course, we are testing on beneficials like bees and monarch butterfly, so that the candidates are not active there. So we have actually less, at the end, less a fraction of the organism spectrum we are looking into are our target organisms, and the major size are our off-target organisms, which makes it very hard to identify new candidates. Of course, there are other factors like insects and diseases and weeds. They have to be non-resistant against this active ingredient, because there is a lot of resistances already in the field. The MOA has to be innovative. That brings in 10 years really innovation, because if it's today obvious and not so one, so Raja Raman, the lead of small molecules since today, she says always ignore the obvious, because if it's today obvious, it will be in 10 years when the active comes into the market, it will be already out-fashioned and not competitive. So it has to be a very new innovative mode of action. Also, with mixtures and formulations, we have also adjuvants that help the actives to penetrate the cell of the insects, for example, but not the crop or other organisms. Of course, we need to find out what is the future market portfolio in 15 years or 14 years. synthesis and scale up, as well as cost of goods for production, is in crop science also quite different compared to pharma. Since in pharma, you need just to take a small pill. In crop protection, you need to spray hundreds of hectares, maybe thousands of hectares land with this compound, and it has to be still worthwhile for the farmer, cost-wise and investment-wise in general, of resources too, also human resources. And also it has to be, of course, for us of added value. Regional conditions and testing. A human in Germany would be probably the same human from metabolic perspective in France or in the US. But when it comes to crop protection, regional conditions in Africa or in North America are quite different. So we have always to test these candidates everywhere, and depending on the results, positioning the products accordingly. As you can imagine, with these all activities, there is a lot of data coming out, a lot, believe me. And we need a lot of data science and data scientists that drive these activities. So we have chemistry labs, we have labs that do high throughput screening, greenhouse testing, and field testing. So when we have the early screening, then when the compound is active, it goes into the greenhouse under predefined conditions. And when there it performs too, it goes then into the field testing in different regions. And we have also the biology around the organisms that I mentioned, the target and the off-target organisms. So there is a lot of data, as mentioned, coming out. And in data science, we have data science teams across the entire organization. Mainly they are cheminformaticians, statisticians, and bioinformaticians. And they use a diverse set of methods and tools to drive each step and at each level better decisions at the end. And of course, digital transformation is a key driver here, because only if the labs, the greenhouses, and the fields are generating data and storing data in the proper way, we can make sense out of data. I think the term shit in, shit out is very common in data science. So it's very important to have proper data stored in the IT infrastructure, in the databases and applications that we can access and integrate and put together from different sources and make model them and make optimally predictions based on them, to also experiment and do the optimization cycle with our labs. So for example, in chemistry, we test few compounds. I mentioned 100,000 compounds, actually it's not few, every year. And when we see, for example, compound is active but not performing as well as we want to have, so we go into this optimization cycle with the cheminformaticians, statisticians, and even bioinformaticians to model and do the virtual screening. I come back to that. Especially here in the bioinformatics, where I'm more focusing on, it's very important for us to understand the power tools, power technologies that we have to generate data that are helpful. And when we talk about data in bioinformatics, we are speaking mainly about genomics, transcriptomics, proteomics, metabolomics, and phenotyping data, which basically we are trying to by integrating all these data, getting better insight into complex biological systems. in the target organisms, but also in the off-target organisms. And in crop science, as mentioned, it's not that easy. We are dealing, as mentioned, with diverse organisms which have different sizes of genomes. And these genomes need to be sequenced, processed, assembled, and finished, that we can identify the target genes of interest, but also understand the activity across these organisms. Here you can see, for example, we start with the viruses, which have the smallest genome size. We move to bacteria, fungi, algae, worms, insects, birds, mammalians, and humans. And then we have plants. With plants, we have the crops and the weeds, which are also, of course, plants. So especially in this field, it becomes even more difficult to get proper genomes. That's why we are relying on our sequencing group located in St. Louis to help us with sequencing all these genomes across all organisms. Here you may see one interesting outlier here with the fungi. This is actually soybean rust, which is the most devastating disease, especially in Brazil. It leads to yield loss of up to 90% if not controlled. Management cost for farmers exceeds $2 billion per season in Brazil alone. The pathogen is highly adaptive, but the number of practical solutions for controlling the disease is limited. So to accelerate innovation here, we develop new traits, novel mode of action to get here better solutions. But it's the most difficult organism we had as far in disease control. This highly complex genome, it is 60 times bigger than the yeast genome and composed of 93% of repetitive elements that you can see here. And it consists of 2 nuclei. Especially here, the lines, I'm not sure if you are aware of circus plots. These basically are the 20 chromosomes of the soybean rust. And you see so basically when you find here one repetitive region that overlaps with another region on another chromosome, here's a line, and you see basically there are a lot of repetitive regions. So analyzing such a genome and building up the genome sequence of such an organism, it's a task for itself. So we are working here also in consortium with external partners and even with competitors to get the genome. So we start with this as a basis for identification for new mode of actions. Here you see also such a impact of soybean rust when it's not controlled by agrochemicals. You see directly, even from the airplane, the effect, or even the satellite, not even airplane. So as mentioned, we need advanced technologies that helps us to move faster with the sequencing and generating genomes of such hard to get organisms. One of the diverse set of technologies, we need to combine them all, and the outcome of all sequencing technologies to get the genome is the nanopore sequencing. So the nanopore sequencing we are using for the diverse set of use cases, not only for the genome sequencing. It's quite interesting technology that allows real-time sequencing of DNA. It's the algorithms behind it. The AI-based algorithms are coming back from voice recognition. So basically the algorithms that are used, for example, for Siri, is basically more or less the same than is used for DNA sequencing. So we start there with these nanopores, which are sitting in the sequencer in the flow cell. We have there hundreds and thousands of these pores that are sequencing in real time, and the base calling is generated across all pores and then aligned to references, for example, here to identify mutations that lead to diverse effects. For example, here we can identify the A as a mutation in this genome, in this one genome compared to a reference genome. The variants, based on these data, we do basically filtering of all the data using variant calling and variant annotation. This is not rocket science in the meantime. It's been used in academia too since some years already. And we are using it also internally to develop our tools and methods for diverse use cases. I want just to make clear here that you hear always, yeah, here we are doing AI, but this is nowadays, I would say, not even innovation, because we are doing it at each step of our work. And without machine learning and AI, we wouldn't move today step forward. So why do we need such good genome sequences? Because then we can do forensics, basically using in silico analysis to predict basically the selectivity of the compounds against specific organisms. Here's just an illustrative example for one target, one mode of action, beta-tubulin, which consists of different subunits. And we scroll through the entire genome set and see which, so how diverse are these targets across these organisms. And some organisms have specific subunits, others have none, others again have twice as much of specific subunits as others. We need to identify also via virtual screening of and virtual docking of the active ingredient to the target molecule, where it's binding in the active ingredient and where is the binding site. Is this binding site existing in our target organism? Is it existing in our off-target organisms? How specific and selective is our product? Also, we are using this information, even if it's not 100% selective, we can use this information to optimize the chemistry, to increase the selectivity, but also the activity of the compound. So this is another interesting example of how we use bioinformatics, this time for the safety by design approach, which we are doing in collaboration also with our pharmaceutical division. While they are focusing on activity in human, we are focusing in non-activity on human. So we are using here AI tools and models to basically compress and translate the chemistry into a grammar, a Smiles grammar, that's a proper one. So there are different solutions for develop AI solutions existing in other industries as well as academia for translating chemistry into machine readable grammar. but not each one is optimal. So the accuracy here is very important. And here we have generated, we are using actually the L1000 data for this publication, but we are using it also for internal, internal compounds, where we basically for selected compounds here, 20K compounds, we have, just as an example, we generated gene expression profiling data, where we basically try to understand what is the expression profile of these compounds, whereby we know which compounds are leading to a toxic result and which ones are leading to non-toxic effect. So thereby we can train the algorithm and the models to generate completely new compounds based on a desired gene expression profiling. So expression profile that doesn't harbor a tox effect. And basically we optimize the chemistry to fit into the desired safety by design approach. And we validate, of course, these learnings, these compounds through the experiments, and use the results again to optimize the training data and the prediction. So let me make here a break and move to another topic. As mentioned, I'm leading innovation projects in data science. I can't speak about the project itself, but I can speak about how to manage innovation projects in data science. So innovation by itself is quite diverse. It can be small and incremental, but it can be sustainable or even disruptive. It helps to identify where you would put your project in this plot. Is your project small and incremental? That's not bad. That might be even easier to get faster with. Or is it disruptive? Depending on where you put your project into that plot, that has different implications. And the environment is also different. You need to consider potentially another different investment options, like for example, around disruptive, we have in some less investment budget, since these cases are rare. But with incremental and sustainable innovations, you have higher budget options, because in some, so there is higher competitive environment, but higher option chance to receive budget. For the disruptive part, you need to convince a lot of people, and you can expect more pushback actually, because people, because especially disruptive projects might include a complete change of processes that people are used to since years, and people usually are afraid of innovation. That's the human nature. The supermarkets next door changed the position of things, and I was messed up in my head. Where are my groceries? So, and I became angry why they have changed this, but they have to optimize, become more efficient. So it's crucial to be not also, if you get pushback, not to stop it. So someone told me, if no one told you that you are crazy with your ideas, you are not in a disruptive field. So yeah, that's important here to find out where you are. Maybe just as a disclaimer, each project is different. As mentioned, it depends on various factors, including the topic, the goals, the customer group. Is it only internal project, or does it involve external? Because that's another level for itself. You have security holes of information and data. That's a big topic. Then you have to find out is the size of the stakeholders when you even internally. Is it only local in your country or even on global scope? Time difference is also difficult here when you go on global scale. Legal and IP implications, timeline of implementation, and of course, change management, how to manage change for the organizations where you bring innovation in. It's, I have to say, not comparable to academic projects. I led already academic projects, and it's quite different. The complexity level is another factor, and you have to maneuver through a lot of obstacles. Yeah, the scale is just different, especially when you go on global level. And what I'm going to present in the next slides is based on my own experience. So I want to start basically by telling you some story here. My first task at Bayer was that my line manager told me, basically more or less on the first day at Bayer, go and do process and data flow mapping. And I told him, to whom, why, me? I am new, I have no idea how the processes at Bayer are working. Don't you? So I had a colleague who is since a long time at the company, and I told him why. So this person would be perfect for such an activity. And he told me, no, I need a fresh idea, a fresh view on all activities, so an unbiased view. So I traveled to the R&D sites globally and talked to people to understand what they do, how they do things, and what are the challenges they are facing in generating data, consuming data, storing them, and analyzing and reporting them further to the next group who is consuming them. Often, what I have realized that there are overlapping changes at a specific step between different groups. And I needed, also what was surprising is people were facing challenges, but humans are adaptive organisms, and often challenges become the new normal. They are tired of it, but they get used to it. So they don't see it anymore as a challenge, and a challenge that might become solved. So often, even at the beginning, there was no solution for it. Time changes a lot, and maybe today there is a challenge. There is a solution for that. Also, it helped also to see all the different. So we had, for example, workshop with the team leads where they shared. So I showed them, I mapped all these data flows and processes where the data comes in, where they are stored, where it goes, what's the next step in the process, and where the bottlenecks are. And gave this map to each group lead and told him, present your issues to the entire, to the other team leads. And everyone presented what they are doing, and they were all very surprised that they have overlapping challenges, but they didn't see it, that the other lab has similar issues. So it was also for the higher management very interesting to oversee the challenges, harmonize the challenges, and prioritize them. So for me, it was also a big learning at the beginning, and that I'm profiting still today, 4 years later, to learn the entire R&D machinery at Bayer, and to get to know a lot of great minds at Bayer. I could build a network within Bayer, which is also very important for my career, I hope. So yeah, this is important. However, you have to, when you identify a challenge in data, either data processing, data consumption, analysis, and so on, you need to find out, what is the source of such a challenge? And if the solution that you have in mind is a sustainable solution. Because if you solve only this small, for one business challenge, find a new solution, that doesn't mean that you have a sustainable solution. You need to basically do optimization cycles of understanding a business challenge, asking why this is the case, finding the right solution, and often you end up with another business challenge that you are facing, and then you go basically these cycles till you get to the root of the actual issue. And by solving that root of the actual issue, you solve basically sustainably for a long time the solution, the challenge, and you might solve other issues that you don't have in mind. So also, it is also important. So to find the solution is not done with it. So you have to basically communicate and get the stakeholders from various collaboration partners internally on the same table, to communicate also the business related advancement that can be made by the solution, by the model you are bringing. And often it's not about the model, it's also about the IT infrastructure around it. It's about the change management of resources, of people that drive this change. So it's not only you need this model to bring in and that's it. Also to leverage the diverse diverse skill set of people in the relevant fields, and to have this sustainable solution. One important factor here is expectation management. So this is a very difficult field to maneuver through. You have to get the right balance between over and underselling, because if you oversell things, you might end up promising things that will not happen, but by underselling you might not get the budget for, because it's not innovative enough, or it's not changing enough, so it's changing to the better. So, but also consider the evolution of mindsets change. So, for example, project that I'm dealing with, I have an idea in mind where we want to be at some point, especially on a data science level. But I don't communicate it directly this way, because a lot of people will get afraid of, and they don't see how to get really, especially if they are not data scientists, how to get from the current status up to that end goal. They think that's unrealistic, and then they not buy into your project. So I tell them the next step, and then showcase them the result. And then through the discussion, we see, oh, we can come to the next step. And then we move stepwise to the next steps, towards the final goal. Yes. So before starting any project, a project plan is the most important part for being well planned, and to have a less risky project run time. So one thing that I learned at the beginning was basically at Bayer, how to start a new project, and that's a process by itself. So that takes also time and investment in this direction. You have to prepare as thoroughly as possible and align the project as thoroughly as possible before pitching it even. So it's very important at the beginning to have what a colleague of mine, Aaron Harris, calls it the burning platform. So what is the current future risk of or impact from business perspective, not from your own work perspective or from your own model perspective, from a business perspective? What is the impact if we not do this project? So imagining it as a burning platform, and you need to bring it down. So from there, where should we go? So what is the future vision of the project? Optimally in 5 to 10 years, what is the end goal? I said at the beginning, it's important to be careful how to communicate this. But if you want to get budget, and especially for the higher management, you need to have a plan. So basically, first a vision, where do you want to go to? And then an implementation strategy. So stepwise business implementation strategy and roadmap. So, and potentially the option to have open for use cases, but also to focus on one, at the beginning on one use case. You can also move from a minimal viable product, an end-to-end minimal viable product with a minimal set and requirements to have a prototype to showcase, and then to get potentially additional budget to add on. Also, a project planning requires work packages and work streams planning, and to get to define what are the required resources, people and budget-wise, especially when we talk about new projects and new innovative projects. That comes often on top of the full-time job of people, and they need to understand why they need, why they should invest in this direction. So in the counting in from the beginning additional resources and asking for additional people, maybe permanent, non-permanent positions at the beginning, but at the, later maybe they get, if the project flies, maybe they can remain. It's very important. Also the budget. It's very difficult to plan budget from the beginning, but it's important to plan as accurate as possible. And then for the project pitch, you have to find sponsors that delivers this budget for the people and money-wise for the technologies that is required for running experiments internally and externally, for services also externally, that brings in added value to the project. And then to pitch this project in front of the sponsors and steering committee. So it's an entire process for itself to pitch the project. But as long as you plan it properly at the beginning, and take time at the beginning, and takes longer time than I expected at the beginning, and it might be frustrating, it's worth it. And one challenge that we often face in innovation projects in large industries like Bayer is that large global companies are driving most of their activities in standardized processes to optimize also automation and overview, for example, for legal, procurement, or IT processes. So the challenge here is how to overcome these standards and drive innovation projects in, with new processes in largely standardized organization. Here it's important to communicate the business impact and to get stakeholders from required organizations on the table to invest in this direction. As mentioned, planning additional resources is always helpful and also helps in convincing people, and finding allies in different organizations to speed up the tasks is important. Also, the sponsors are very powerful, and the steering committee are mostly high managers, and they are powerful, and you can use their power by communicating very early any hurdles to them to get things moving faster. So despite standardization, there are options for moving forward, but also large companies know that this is an issue. That's why they are focusing on methods and developing methods, how to drive innovation actively. Such solutions or methods we have at Bayer, generally not only in crop science, so R&D Bayer-wide. We have different programs like the R&D Tech Immersion, where we go with academia together in, I don't know, a month, 2 months, and work on specific business challenge and try to find a solution. Life science collaborations. So this is an interesting program where anyone at the company can pitch a project, innovation project to the organization. We have a platform that helps us, basically everyone can invest in these projects, depending if they, if you think they are worth it to invest in. It's virtual money for the beginning. And then when, so the projects with the highest investment from the entire global organization and people, they get, funding, real funding, money and people-wise to drive their projects for several years even. Yes, so there are also innovation projects on external collaborations like the Life Hub programs. We have a Life Hub in Boston, in France, and in Leverkusen. Yes. And we have rapid innovation sources like the Catalyst Fund and Catalyst Box. We have the WeSolve platform, which is very funny and so not funny, it's fun platform where everyone at the company can pitch very briefly, oh, I have this challenge. Can someone help me at Bayer? And basically up to 10,000 people who can respond to you. So also going to towards social innovation, like smallholder farmers. This is one of my favorite activities, since smallholders are providing actually 97% of the world's 80% of the food in developing countries. So how to support these smallholders, not only by providing them the right products, but how can we help them for build up collaborations and to drive their farms, and also giving them loans. So there are a lot of options. However, still, when you what I learned, interestingly, Liz mentioned that I am leading the EMEA, so the European network for women data science. These networks, building up such networks within your organization or joining communities, helps you also to connect with people globally, and then you have access points to people, how they can help you in these projects. So in the Women Data Science network, we have the vision to create inclusive culture to empower women and enhance their ability to shape data and data science at Bayer. We have 4 key activities: inspiring by connecting women with innovations and role models in data science at Bayer and beyond. And that, of course, I learned and met people from Singapore to Wessex to San Francisco. So all over the world, that people that I might have never talked to and never learned that they have similar use case than my project I'm working on. So we are attracting also diverse data science talent by continuing to nurture a vibrant, supportive community for women data science. We are championing women also internally and externally by focused publicity and recognition. So that is good for this distinguished women data science, but also for the next rounds of champions. And we grow with that the literacy at Bayer. For example, we are giving trainings to non-data scientists, but also to other data scientists in programming languages, but also in career panels and in communication and so on. So also the Distinguished Women Data Science Awards are very interesting ones that are, so we have great feedback around it. And we are doing a lot of these activities currently virtually and having fun together. And therewith, I would like to thank you and basically close my presentation.
Arwa, thank you. That was such a wonderful talk. And I am incredibly impressed how you gave us real insights into some of the complex data science and genomics work that you've been working on with your colleagues, but also managed to take those insights and come up to a higher level to talk about data science innovation more generally and your personal experience and what you've learned in your time at Bayer. So thank you so much. It was a wonderful talk. We do have a few minutes for questions. And much like your talk, actually, our questions, some of them are very narrowly focused on specific scientific questions and others are more general. So I'm going to, if I may, start with some of the more specific questions and back up right to the beginning of your talk. So the first question that came in was from Ravi Shastri, who I think is still with us. We do tend to lose a few people at the top of the hour. And he asked, how has AI and machine learning reduced the time from 12 to 14 years? And I think what he was asking is in the context of compound development. So you talked about the time it takes to develop a compound costs 300 million euros and it's 14 years. How have you seen efficiencies develop with a reliance on new data science technologies?
Exactly. So this is a very good question, since in the past, the crop protection field was mainly based on the spray and pray way. But in the last year, it has changed, especially due to these advanced data science tools like machine learning or deep learning, in general AI. So these activities around early screening of compounds at the beginning, before even testing them on any organisms, helped a lot. So prior to screening, we are testing the compounds we have. We are optimizing the compounds with our chemists and biochemists together to address the desired properties. But also, for example, this tox related safety by design approach that we are using, applying using AI, helped a lot in reducing, for example, unnecessary animal testing, which takes also a few years. So that we need not only test the first generation, but the second and third generation of animals, and that reduces also the number of animal use, like rats, which is an important target for us. In general, it reduces a lot of activities and helps to focus on the most the candidate with most potential towards success and shorten it a bit. Currently, we are still not there to say shorten it 2 years or I am not sure how much exactly, but for sure a lot of it shortens a lot of activities, individual ones.
Excellent, thank you. The next question we had is also fairly specific. It's from Kenneth Chen, and he asks, how do you approach monitoring many models, and what challenges or specific areas, e.g. performance, drift, explainability, bias, etc., are of most interest? Are there certain kinds of model types that present more challenges than others? For example, CV, tabular, recommender systems?
Yes, so the most challenge we have currently is data cleaning at the beginning, basically. So that's challenge not only on the academic side, but also in the industry. So that's why digital transformation at the beginning, that's the labs, the greenhouses, and the fields all over the world having a solution that where they enter the data properly before we start doing data science. And integrating this data from different sources all over the world is also one of the biggest challenges. Evaluation of models is more on the hands of the experts, of the machine learners, deep learners, and so on. So, of course, aligning these models also across the world, we have R&D sites across more than 50 countries. So that's a big challenge that we didn't solve yet. It's work in progress, I would say.
Excellent. And building on that, a question that just came in, and then Nicholas, I'm going to come back to yours. Sunil asks, could you help us understand a little more how learning from pharma and experience in pharma at Bayer has helped crop management models? So I think it's been fascinating as you've talked to look at some of the differences between the pharmaceutical development and the crop science division, and especially how much more complex the crop science division is, arguably. But what were you able to learn from your colleagues in pharmaceuticals at Bayer on the crop science side?
So I think shortly before I joined Bayer, I think 2016, 2015, before that time, Bayer, pharmaceuticals, consumer health, and crop science were under the Bayer umbrella, but they were individual legal entities. Mm-hmm. So sharing data across pharmaceuticals, crop science, and consumer health wasn't so easy, because there are also third-party compounds which need to be anyway taken out, and the entire database needs to be cleaned up from the third-party compounds to be shared. So sharing the not only the compounds but also the testing results across pharma and crop science helped us a lot in finding common solutions on not only on the compound perspective, but also finding new mode of actions that are interesting for both organizations, or maybe especially in that case for pharma didn't work out because it's not that innovative, or it's not active enough on that mode of action. But for us, it's good because we know it's not active on human. Yeah. So probably it's good to have it to test it on the compounds and on the target organisms we have. Also sharing models, basically challenging each other around the models we are developing. Learning in that course helps a lot for sure in both directions.
Thank you. We are almost up against time, and I don't usually finish with a very, very specific question, but I have a feeling this is one that Nicholas will want answered sort of to scratch that itch. So he says, what was the neural network looking structure on the nanopore page helping with? It looked like it was involved with multiple steps, but I don't think I caught the explanation. Are you able to quickly answer that?
So I'm not sure if that answers the question but we have so basically multiple steps of neural networks that we need to run. And as mentioned, this is not developed by us. It's developed by Oxford Nanopore with other collaborators. So the first one would be basically using these basically the interruption of the electric current, which is happening depending on what phase is running, so AC or G or T running through the nanopore, and translating this interruption of current, electric current across hundreds of nanopores. This is only showcasing one nanopore outcome. For this we use base calling neural networks to basically generate the DNA sequence out of it. Then we need to do the entire alignment, and then we need to generate variants and identify mutations across millions of reads that are generated. And there are often also mutations found somewhere else, but they are probably of less quality or not occurring as enough. Maybe they are due to accuracy mistakes by the first neuronal network. This one we correct with the second variant calling neural network, and basically annotate what this mutation leads to, for less selectivity, less activity, and so on.
That makes sense.
That answers the question. I hope so.
I hope so. I hope that answers the questions. I think it does, because I think we're seeing the neural networks come into play in the base calling and variant calling stages. And so I think we are right at time. So with that, let me thank you again. This is, as I mentioned at the beginning, this is the last industry seminar of the year for us. We will be back. Right now, we have our first seminar for 2021-2022 scheduled in October. We may sneak one in September, but this has been such a lovely way to end the year. Arwa, thank you so much. And I hope you enjoyed the rest of your day, and folks online, thank you for joining us, and enjoy the sunshine if you're here in Cambridge or nearby. Arwa, thank you. Bye, everyone.
Thank you. It was a pleasure. Bye.